Hello, everyone. We're going to get started in just a minute. We'll wait a second to let everyone into the Zoom. Happy New Year, everyone. This is Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. Welcome to the first ASCCT-ESTIV webinar of the new year. This webinar, as all our webinars, are presented by the European Society for Toxicology in Vitro and ASCCT. And I want to encourage you to visit our website and consider becoming a member to support the programs like this that we put on, as well as meetings, awards, and other activities. This webinar is recorded, and the recording will be posted on the ASCCT website in a few days. In the webinar platform, you can ask questions of the presenters using the Q&A feature. Please, at the bottom of your screen, use the Q&A feature to ask your questions, and you can add them at any time. We'll have a short Q&A session after each presenter. You can also vote for questions that you see that you'd like to hear the answer for if they're similar to yours. We've also enabled closed captions, and you can turn them on or off in the toolbar if you like. And then please say hello, Happy New Year, and let us know where you're joining from. Before we get started with the presentations, I just wanted to do a couple of brief announcements. First of all, ESTIV will be having its 23rd International Congress in Maastricht in 2026. That's going to be early summer. And they are open for session proposal submissions until April 1st. So go ahead and go on over to their website and check that out. Also, ASCCT will be having a meeting this year in October 2025. Stay tuned. It's probably going to be in the DC area. We're finalizing the dates and the venue right now. And we will open up session proposals soon. We have several webinars coming up this year, many of them featuring winners of awards from both of our annual meetings, like this one today. If you want to visit our website, you can see on the main page, we have a calendar that's always updated with ASCCT activities as well as partner organization activities. The next one is going to be for February 20th, and it's featuring 3 speakers from the ASCCT award winners group. And so today we have 2 presentations. The first presenter is Giulia De Negri Atanasio. She is currently a postdoctoral student at the Department of Earth, Environment, and Life Science, University of Genoa, Italy, in the group coordinated by Professor Elena Grasselli. She's currently involved in the Horizon 2020 Eco-Efficient and working on the biocompatibility tests. Her work is focused on the study of skin and intestine physiology related to ingredients or formulated products in the cosmetics and nutraceutical field. Our second presenter is Kim To. She is an environmental statistician at ICF supporting toxicological and environmental health data analysis. Prior to ICF, Dr. To was a principal biostatistician at Innovative, where she supported the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods through computational tool development and leadership of the integrated chemical environment. So I will now turn the floor over to Giulia, where she'll talk about the evaluation of physiological repeated exposure of aluminum in a 3D intestinal tissue model.
Thank you very much for the presentation. I'm going to share my screen. Okay. Can you see it?
Not
yet. Now?
Yeah, it's coming.
Okay. So, good evening, everyone. Thank you to give me the opportunity to be here to present my work that I have already presented in ESTIV, which is about the evaluation of physiological repeated exposure of aluminium in a 3D intestinal tissue model. So I am Giulia De Negri from the University of Genova in Italy. So aluminium is the third element which is present in the Earth crust, and it is a very versatile metal which is found in several consumer products from makeup and skincare to food packaging and drug formulation. And due to this large application, it is important to investigate the impact on the human health and the possible implication. There are several routes of exposure to aluminium. The first one is via inhalation, and aluminium particles which are present in the air can be inhalated and contribute to the absorption into the body. The second route of exposure is via injection, where some pharmaceutical drugs can contain aluminium salt, which are used as adjuvant additives. The third route of exposure is via contact, where aluminium, which is present in the several cosmetics, can be absorbed through the skin. And many cosmetics, such as lipstick and antiperspirants, can contain aluminium, which is used for its properties. And the last route of exposure is via absorption, and usually from the 0.1 to 1% of indigested aluminium is absorbed by the intestine. And aluminium can be absorbed not only from oral drugs, but also from solid food and drinking water. Indeed, aluminium is present in the food naturally, for example, in vegetables and cereal, but it can be also added intentionally, used as food additives, or it can be leached by the packaging, for example, from foil wrap, cans, or it can be leached by the cookwares, for example, from the pans. In particular, if we have a acid food, we can have an increase of leaching aluminium in the food from the packaging or the cookware. The prolonged exposure to aluminium can have some physiological effect on the human body. For example, it can have effect on the nervous system, which is associated with cognitive decline, including memory loss and decreased problem-solving abilities. Moreover, it can induce an oxidative stress, which can lead in a cell damage. Furthermore, the aluminium can induce some bone and skeletal disorder, which are linked to the development of osteoporosis. This condition is characterized by a reduced bone density and an increase of fragility. Moreover, it can induce problems at the kidney. Indeed, aluminium can accumulate in the kidney, and it can interfering with the ability to filter and increase the risk of the development of kidney stones. So the aim of our work is to study the repeated exposure of aluminium and investigate the consequence of this repeated aluminium exposure on the structural and functional integrity of the 3D intestinal tissue model. We use a 3D human intestinal tissue model, which presents a complexity that can capture the multi-layer structure and the dynamics environment of the human gut, which can offer a more realistic platform to studying the aluminium absorption and the toxicity. Moreover, this 3D tissue model presents several advantages which can help to investigate the physiological response, such as the barrier function and the nutrient transport, in a controlled and reproducible way. The 3D tissue model presents several applications where the This model can be utilized to evaluate the impact of the repeated aluminum exposure on the intestinal health, and this can help to underline the mechanism of the aluminum-induced toxicity. So our experimental plan was as follows. So we have our 3D tissue model, and we investigate 4 different concentrations of aluminum. The 0 ppm represent our control, and then we have 3 different concentrations of aluminum, 5, 20, and 50 ppm. We give to our tissue 12 days for 12 days this concentration of aluminum, and during all the experiment we keep the tier which give us the information about the activity of our tissue. Moreover, at the end of the experiment, we investigate the total amount of aluminum which surpass the layer in order to investigate the amount of aluminum that can reach the bloodstream. To do that, we use the ECP technique, so the inductively coupled plasma. Moreover, we study some gene expression and the morphology of the tissue through the hematoxylin technique, and we investigate the microvilli morphology using the TEM. So the first results are reported in this graph, and we found that all the tissue treated with the different concentration of aluminum present more or less the same trends. Just the tissue treated with the 50 ppm at the 3 last days present a slight different. Indeed, here we have a zoom in just of the last day, so at the 12 days, and we found that the tissue treated with the 50 ppm present a statistical difference of the transepithelial electrical resistance. So at this day, something happened to the tissue, and we are gonna to explain after. Moreover, we investigate the total amount of aluminum found in the donor compartment, which represent our bloodstream. And we found that there is a correlation between the total amount of aluminum that we put on the top of our tissue and the amount that we found in the bloodstream. Even if the total amount of aluminum that we found at the 2 higher concentration is not really high compared to the amount that we put at the beginning. Here we have the gene expression of some gene that we studied. We have the metallothionein. In particular, we studied the isoform 1E. The metallothionein are proteins that can bind the metal and have a role to protect the cells against the toxicity of the metal and the oxidative stress. And indeed, we found that the tissue treated with the highest concentration of aluminum present a higher expression of this protein. Moreover, we investigate about the occludin and claudin, which are the tangential integral membrane proteins. And looking at the occludin, we have a decrease of expression of occludin just in the tissue treated with the highest concentration. While looking at the expression of claudin, we did not found any expression at the tissue treated both with 20 and 50 ppm. Moreover, we study the morphology of our tissue, and at all the tissue treated with all the concentration, we did not found any significant difference. Just the tissue treated with the 50 ppm present some cells with a vacuolated aspect, which could indicate that the treatment could trigger a detrimental effect after the 12 days of higher concentration of aluminum treatment. Looking at the image that we obtained through the TEM, here we have two representative images. The first one is our control, and the second one is the tissue treated with the 50 ppm. We can see that we have a different morphology of our microvilli, and moreover we measure the height of the microvilli, and we found that there is a correlation between the amount of aluminium that we put on our tissue and the microvilli height. In conclusion, we can say that the result of this study suggests that the repeated exposure to aluminium at the tested concentration could lead to some detrimental but not destructive effect on the 3D intestinal tissue model, only at the higher concentration that we studied. Nowadays, some other research are ongoing in order to explore any potential long effect and to understand the possible implication. So I have concluded, but I would like to thank all of my group from the University of Genova, and in particular my PI, Professor Elena Grasselli, and then two companies that we are working on, so Angel Consulting and Mattec Corporation, which provide us the tissue. And of course, I would like to thank all of you. Thank you very much.
Wonderful. Thank you, Giulia. There
Thank you.
are a couple of questions coming in, so I will start asking them for you.
Yeah.
The first one is, did I understand correctly that the amount of aluminum measured at the end of the experiment is not the same as the amount of aluminum that was administered?
Yeah. Yes,
Okay.
it
And
is
do
exactly.
you have an idea of why that might be?
We think that maybe something microparticle or nanoparticle are inside the tissue. Indeed, when we did when we observed this tissue through the TEM, we found something, but we would like to investigate better this aspect.
Okay, great, thank you. Just one clarification question. Did you use a commercially available 3D model?
Yes, the one from Matte Corporation. Yes.
Okay.
Yeah. They provide to us them, and we use that
Okay.
tissues.
And then in addition, could you provide an explanation on the rationale for the concentration and the times used in the experiment?
So the concentration that we use, the 5 and 20 ppm of aluminium, we can find them in some vegetable or, for example, in mushrooms. And while the 50 ppm is a concentration that we decide to use just to arrive, at a very big concentration of aluminium. And we decide to use 12 days because it could represent something that, I don't know, maybe someone can eat every day, some lettuce or salad. So we would like to find quite long times, but not too long, because otherwise the tissue could, come back older and the result could not be all very good.
Okay, thank you. Another question, how do you explain
Yeah.
How do you explain that you have measured aluminum on the other membrane side at the highest dose? So I think they're asking how the aluminum got through the tissue. Is it a transporter, or is it
I think that something happened with the tank junction, and we found something with the PCR. Nowadays, we are gonna to do the Western blot in order to see if we have a concrete difference between all the concentrations. So I think that is something in this protein pathway.
Okay. They followed up with another question, which is, do you think it's related to the downregulation of the occludin protein?
Yeah, I think that, but I'm not sure, but we will,
Okay.
but I think this is something linked to that.
Okay, great. I have one question for you, and I was wondering, towards the end of your presentation, you showed that the microvilli, were they shortening in length, or were they just fewer microvilli? I wasn't sure.
So they were shorter and they were less.
Both.
So
Okay,
there are both. Yeah, exactly.
okay,
And
great.
moreover, the morphology of the tissue was not the same. It was like quite different.
Okay. Oh, that's interesting too. Okay, yeah.
Yeah.
Lots of things to follow up on to figure that out. So there's one more question
Okay.
coming in. Your study involved constant exposure, but in vivo, there might be fluctuations, and it won't be that high constant level of exposure. Do you think that would affect the results if there are higher and lower, amounts of exposure?
Uh, so now we are doing this result in order, to have just we are in the best situation now. So we are doing something in vitro, so it's easier. But I think that in vivos it's different because it's not all the same, the amount that you digest with the of aluminium. So I think that it could be not the best representation, but we starting to study that just to have the best situation, in a much controlled way.
Yeah. There might be
I
a
don't
way
know if
to study the fluctuations in vitro. You would have to
yeah,
remove
we could
some
do
and then
that.
add again, maybe. I don't know.
Yeah, it's a good point. Yeah, we can do something like that, actually. Yes, good point. Thanks.
Okay, we're going to move on to the next presentation to Kim To. Kim, if you would like to share your screen.
Yep.
Thanks, everyone, for all of your questions, and thanks very much, Julia, for your presentation. It was very nice.
Thank you.
Okay,
Okay.
slides look good, so I'll give you the floor.
Okay. So I'll be presenting the topic of my ASCCT poster from last year's annual meeting, DASAP version 2.0, implementing OECD Guideline 497 updates. Here's a really brief outline. I'll give some background materials. I'm going to go over two of the major updates in DASAP version 2, and then, time permitting, do a live demo. So just for background, here's a screenshot of the DASAP and a URL in the top right. This is an open source web application for users to apply defined approaches for skin sensitization to their own data. And I've underlined the terms defined approaches and skin sensitization here, and I'll talk about both of those next. Here we have the definition of a defined approach, which consists of a defined set of information sources and a fixed data interpretation procedure to derive a prediction without the need for expert judgment. So essentially, this is going to lay out exactly where you should be getting your data from and exactly how you should be analyzing it in order to get, hopefully, a high confidence prediction. So the endpoint that we want to predict is skin sensitization, and this is an endpoint evaluated in hazard and risk assessment of chemicals, and it occurs in two phases where ultimately in that second phase, exposure to that skin sensitizer is going to lead to allergic contact dermatitis. This endpoint has traditionally been evaluated with the in vivo murine local lymph node assay or the guinea pig maximization test. And more recently, defined approaches for skin sensitization have been developed. And these leverage the adverse outcome pathway for skin sensitization in order to integrate data from non-animal assays as well as in silico models. Here we have the skin sensitization adverse outcome pathway. You'll see there's 4 key events and then our adverse outcome. And we're going to be looking at key events 1 through 3. For each of these key events, there is an associated OECD test guideline that describes non-animal assays which map to their corresponding key events shown here. So we have these key events with these assays, and we have this adverse outcome pathway. So with that information, these assays have been used together in defined approaches to derive human relevant hazard and potency predictions for potential skin sensitizers. So more specifically, the OECD guideline on defined approaches for skin sensitization was published in June 2021. This guideline, Guideline 497, formalizes the combination of several in vitro, in chemico, and in silico information sources into two defined approaches, which are the 2 out of 3 in the integrated testing strategy. These two defined approaches are what's available in the DAS app and what were updated with version 2. Going back to our key events, here again, you see the 3 OECD test guidelines and the assays described within them. This time I've separated them. So in the top, you see the 3 specific assays that are lined out in Guideline 497 for use within the 203 and the ITS. And then on the bottom, you have these other assays that are within those OECD test guidelines, but are currently under evaluation for inclusion in Guideline 497 as alternate information sources. So just to highlight, we have DPRA, keratinescence, and HCLAT, which are in the current version. Tying everything back to the actual app, so we have those 2 defined approaches in Guideline 497, and this app really just provides a point-and-click interface to apply the data interpretation procedures from those defined approaches. So in terms of using the app, the user would need to derive their data externally using whichever assays or other information sources are defined in those defined approaches, and then this app will take that data and process it. In terms of the updates, with our first iteration of DAS app, the ITS and 203 defined approaches were included as described in Guideline 497. So just with those 3 assays, the DPRA, the HCLAT, and the keratinescence. With version 2.0, there were a number of updates, but the 2 I'm focusing on are adding those additional assay options for the ITS and 203 and adding borderline result evaluation for the 203. This is something that's also in the guideline that I didn't talk about yet. So going into the updates, first we'll talk about the integrated testing strategy. Again, with a defined approach, you have your information sources and then your data interpretation procedure. With the integrated testing strategy, this uses as information sources a KE1 assay, a KE3 assay, as well as an in silico prediction. And shown in that second column are the assays and the models that are in Guideline 497, and on the right are the ones that are currently under evaluation. For the data interpretation procedure for this defined approach, it's a scoring scheme. So there's ITS scores assigned to the quantitative endpoints for the KE1 assay and KE3 assay. And then you also score the in silico prediction. And the sum of those assay scores dictates what the hazard identification and the potency prediction are using a ITS lookup table. For DAS app version 2, highlighted here are the assays that were added for the ITS. Here's just kind of a picture of exactly what was added. Before, you could only do the DPRA and the HCLAT, and with this version 2, we've added in the scoring schemes for the ADRA, the guard skin, and the USense. Here's just an example of the output that you get from the DAS app for the integrated testing strategy. With the addition of these new assays, we've added in just a little bit more information summarizing the user's selections. And then it also provides the individual ITS scores, the total score, and then those hazard identifications and potency predictions. Next, I'll talk about the 2 out of 3 updates. For the information sources, we've got the KE1, KE2, and KE3 assay in Guideline 497, again, DPRA, keratinescence, and HCLAT. And again, on the right, the assays that are under evaluation. So this data interpretation procedure is pretty simple. It's just a consensus decision. So if 2 of your assays are positive, then the outcome for the 2 out of 3 is positive because 2 out of 3 of them agreed. And so programmatically, that's not really what the update in version 2 was. Really what we did was add a borderline workflow. So for each of those assays, you want to get your overall outcome positive or negative, and that's going to be based on some continuous data, probably defined within the assay protocol, how to get these binary classifications, and using those cutoff values for those classifications. In terms of the 203, if you're using data from those assays and any of them are based on values that are close to those cutoffs, it's going to introduce some uncertainty into that prediction. Guideline 497 it describes these borderline ranges. So in this table, you'll see the cutoff for those continuous endpoints from the assays, as well as the borderline ranges that are laid out in Guideline 497. So when you're running your assays, you would use these borderline ranges to determine whether or not you have a conclusive result. And if it's not, it's a borderline result and can't actually be used in the 2 out of 3. So I said the deeper keratinocytes and HCLAT are described in Guideline 497. There are also cutoff values and borderline ranges defined for those additional assays being evaluated. With the first version of the DAS app, you couldn't run through these little decision trees. You would just provide the overall data and have to do the borderline evaluation externally. But we've introduced these, I think there's 8 of them, new decision trees. And this is just to represent that each tree looks different. So it's just like a stack of figures. Let's see. Just an overview of what the DAS app is going to do for you. You prepare your data according to the template that we've got, upload it, and then select which assays your data came from. And then the app processes the data through each of these decision trees. And then it goes through the actual 2 out of 3 data interpretation procedure to get you those final 2 out of 3 predictions. So just to summarize before we go into a demo, the DAS app enables users to apply the 2 out of 3 and ITS defined approaches as described in Guideline 497. And with this latest version 2 update, we have borderline evaluation and also additional assay options for the integrated testing strategy. So I will go into the live demo. This. So when you launch the DAS app, this is the page. I usually click
Kim?
this launch app in new window.
Hey, Kim,
Yep.
you stopped sharing your screen.
Awesome. Okay, hold on. Got it?
Yep, we can see it now.
Okay, perfect. All right, so this is the landing page for the DAS app. I usually click on this link just because I like having it in one big open window. So you have a little description of the app, and then in this first panel, you have the 2 out of 3, the integrated testing strategy, and then another defined approach that's not in the guideline. There's information boxes next to them, and it'll pull up those diagrams for you, as well as the assays that are applicable to the defined approach within this app. So you can select whichever one you want, and now we have this option to do borderlines for the 2 out of 3. I'll just go over the borderline evaluation demo. So once I do that, I confirm the selection. With this, a data template is provided. There's a worksheet for every single assay that is possible, and it'll show which endpoints are required for running the approach, and you just have to fill it in and put it in there. So I've got an Excel file with the demo data. Click open. This workbook has 3 worksheets, and you'll first see the first worksheet in your file, and then you can use this little drop down to look at the other data. Oh, I should note for the keratinocytes specifically, the assay developers, I think, have already laid out a template for doing the borderline evaluation. So in terms of the data requirements here, it's just what that output from the template is, which is why it's just positives and negatives and not any individual assay data like with the HCLAT. So my data looks okay. I click continue. In this next section, you'll see it's broken up by the information source. So here we have our 3 key events. You have the selection, which KE1 assay did you use? In this case, it was the DPRA. And you select the worksheet that has the data for the DPRA. And then it's going to show boxes for each of the required endpoints. You select the column that contains that information and do that for all of the information sources. So I'll go ahead and just click through those. Just the HCLAT. So we have everything set up with our columns. Click done. The next page lets you review everything that you've selected. So the defined approach you've selected, the type of analysis, the file that you uploaded, and then it goes through each of the tables and tells you whether or not there were any issues with the formatting of the data. So if this is flagged, it'll be orange and bold and say true. The last thing that's done is it checks to make sure that each of your chemicals has data from at least 2 assays, because otherwise the 2 out of 3 can't be done. Everything looks, I mean, there's flags, but we'll just run it anyway. It'll give you a warning. And here is the output. You have the overall outcomes for the individual assays and then the actual 2 out of 3 hazard prediction. In this case, this one has NA because it didn't have enough data. It only had data for HCLAT within the files as part of the demo. And then on the bottom, you will have more detailed results about the assay results. So you have your individual outcomes from each run. And then you can also download this as an Excel file and it'll have all of that data that I showed. There's also a comparison option, but it's not yet available for the borderline results. It is for the standard workflow, but we should be including that soon. Let's see. So here's my acknowledgements page, the NICEDM group, and ASCCT and PhD posters for the poster award. Thank you.
Thank you, Kim. That was a great presentation. I love when we have a demo. I'm sure folks really appreciated being walked through that. I was wondering if you could mention for folks who might be wondering, you said that there is another DA in the DAS app that's not in the guideline. Why is that in there?
This one is included in the EPA's draft science policy. Here it is. There's the title. This very long title.
Yes.
And from what I've read, it is recommended, like these are recommended for use based on that draft guideline, which is why we've included it.
Yes, excellent. Thank you for mentioning that. Okay, we have a couple of questions coming in. I want to tell folks we're going to go ahead and let you raise your hand also. If you want to raise your hand and ask a question of Kim orally, just go ahead and do that. You can do that in the reactions or in the toolbar below. But otherwise, please type a question in the chat. So the first question we have is, could you perhaps use artificial intelligence to pull information and determine the correct defined approach?
I am not sure, just because in terms of the guideline and validating the defined approaches within the guideline, those were based on curated data, both human and LLNA curated data. So those weren't done with AI. I'm sure you could use AI to do some type of lit search to get more data, but for right now, what's being done is just using the curated data.
Yeah, I wonder, it sounds a little bit more like a development question, like if you're developing different defined approaches and trying to decide which methods to combine together, maybe then it would be useful.
Yeah.
But in this case, I think you're putting the defined approaches out that are already in the OECD test guideline.
Correct.
Next question is, can you elaborate on the advantage of including the borderline evaluation?
Sure. So number one, it's been requested from our stakeholders a couple times. And number two, I believe the reason it's been requested, or what I've heard, is just that the borderline it's very important to make sure that you do that before you do the 2 out of 3, because otherwise your results may be low confidence. And so this is just really a way to save time, as well as just create a streamlined process from getting your data prepared for that borderline evaluation to getting your 2 out of 3 prediction.
Yep. Okay, thank you. Is there a flag function if the input is out of the applicability domain of a given method?
No, there is not. That would depend on the user. So I guess when they're doing their experimental setup, kind of going through those requirements of the individual assays as well as the defined approaches themselves.
Yeah, that's more around solubility within the in vitro assays and things like that, which should be within the test guidelines when you're actually doing the tests. Right. Next question is there an intention, maybe this is a future request, is there intention to create a Word document or PDF with the results from the 2 out of 3 that could be used to support single chemical submissions instead of the Excel spreadsheet?
So that is not on my radar, but now it is. And so I can bring it up to our client and try to see if we can do that for sure.
Great. Okay, thanks, Kim.
Yeah.
I don't see any more questions. Sorry. As soon as I say that, of course, a question came into the chat. If you know this, what industries are represented amongst the users of the app?
I do not have the answer to that. That's my answer. Sorry.
That's okay. That's okay. It- uh, Maybe I could say that if you look at we can assume, if you look at the OECD test guideline, that those guidelines are typically valid for chemicals, pharmaceuticals, and pesticides. So I would say that's probably the user base, and then of course cosmetics and personal care products as well. All right, well, I would like to thank you, Kim, for your presentation. I would like to thank you, Giulia, as well. Congratulations to both of you on your awards. Thank you for doing such good science that we can recognize it. Thanks to all of you for joining the webinar today and for your good questions and discussion. And we hope you have a good weekend.
Thank you.
Thanks very much.
Thank you. Bye-bye.